Okay. It's another very interesting time. Tumekutana tena na leo tutaenda kuendelea na topic 2 katika form 1 biology. Tutakuwa nafanya discussion ya scientific processes in biology. Hii topic ya pili katika form 1 biology. So tutaanza discussion yake leo na kuona different things which we need to understand in this topic. In the topic 1 we discuss about the introduction to biology, O-level biology, and we discussed a lot of things regarding the definition of biology and regarding the characteristics of living things. Kwa hiyo ile ndo ilikuwa topic ya kwanza. Now, in this topic we'll be discussing about the scientific processes in biology. What is scientific processes in biology actually mean? What does it mean actually? Katika topic hii tutakuwa na discuss namna ambazo tunaweza kufanya kitu chochote, yani anything which you want to do in biology, in biological studies. In biological studies we need to know a lot of things. Kuna vitu mbalimbali utahitaji kuvijua. So in order to know the things we need to have different procedures in order to collect data and to know informations. Let's say kwa mfano una viumbe fulani ambao wako wengi, na wale viumbe unataka kuwaweka kwenye makundi mbalimbali, unataka kuwa classify. Let's say unapotaka kuwaweka kwenye makundi mbalimbali, lazima ujue namna mbalimbali utawafanyaje ili waweke kwenye makundi mbalimbali. Kile kitendo cha kuwachukua viumbe wengi na kuwaweka kwenye makundi mbalimbali ni mfano wa scientific process. Process kwa Kiswahili cha kawaida ni kitendo, scientific ni cha kisayansi. So scientific processes in biology ni vitendo vya kisayansi katika biology. Kuna vitendo mbalimbali vya kisayansi katika biology ambavyo tutaenda kuviona mifano yake baadaye. Now tunachokisema ni kwamba biology is just like other science subjects. It involves carrying out experiments. Inahusisha kufanya nini? Majaribio. Experiments ni majaribio. So masomo yote ya science huwa involve carrying out the experiments, inahusisha kufanya majaribio. Now when studying living things, simple observation can be made by using our own sense. Unaona, unaweza kufanya simple observation, vitu ambavyo vinaonekana kwa macho ya kawaida. Kwa mfano, you can see, which is sight. You can sense smell, you can see sight, unatumia macho. You sense smell kwa kutumia pua. And then you can sense touch by using skin, or you can sense taste by using the taste buds in the tongue, but also you can sense hearing or sound by using ears. Unaona? Kwa hiyo the senses can be detected by our sense organs. Unaona? The senses is hapa: sight, smell, touch, taste and hearing. They are detected by the sense organs. Sense organs zinashika, zina detect. Kwa mfano, we have eye ambayo ni jicho for sight, kwa ajili ya kuona; nose for smell, pua kwa ajili ya kunusa; the skin for touch, ngozi kwa ajili ya kugusa; tongue for taste, ulimi kwa ajili ya kuonja; and finally the ear for hearing, sikio kwa ajili ya kusikia. Now to use our own sense organs to make correct observation. Tunaweza kutumia sense organ to make correct observation. Now I think we know about the body sense organs, which we started in primary school. We called them the milango ya fahamu in Swahili, but for those who studied the English medium schools, they still called them the sense organs, lakini ni milango ya fahamu. So we have eye ambayo ni jicho, which is the sense organ for vision, kwa ajili ya kuona. It is the receptor sensitive to light, in a sense mwanga wa aina yote unaangaliwa na jicho. And then we have ears ambayo ni masikio, they sense hearing, kwa ajili ya kusikia. So receptor sensitive to sound vibrations, mitikisiko ya sauti, the sound vibrations. Any sound is detected by ear. But also there is the part of ear which is involved with balance receptor. Balance manake nini? Wewe unavyojijua kwamba sasa hivi nimekaa au nimesimama au nimelala, ni sikio ndio ambalo linapeleka taarifa kwenye brain. So kwa mfano, mtu ambaye analewa pombe, the part of the brain which is affected by alcohol is the cerebellum. Cerebellum ina receptor ambazo zinatoka kwenye sikio. Kwa hiyo sikio linapeleka receptor kwenye cerebellum, ambayo cerebellum kazi yake ni ku maintain balance. Kwa hiyo that means if cerebellum is affected, huyu mtu atakuwa hana balance. Ndio maana mtu aliyelewa pombe anakuwa na yumba. So balance receptors are sensitive to change in body position. They are also present in the ear, which we shall discuss it later in Form 3 biology. But also we have the nose, which is sense organ for smell. Nose ambayo ni pua tunatumia kwa ajili ya kunusa. It is sensitive to chemicals, different chemicals. And then you have the tongue, ambayo tongue inatumika kwa ajili ya kuonja. It is sensitive to tasting. It is receptor to chemicals also. Unaona tongue pia ina sense chemicals. And then you have skin, which is sensitive to touch. Unaona sensitive to touch. But not only sensitive to touch, skin senses different things like touch, pressure, temperature. All of these, they are sensed by skin. And I think you know skin ni ngozi, tongue ni ulimi, no, nose ni pua, and then ear ni sikio, and eye ni jicho. Now, these are the sense organs which we use them for sensing different conditions in our environment. But kwenye topic inasoma nini? Scientific processes in biology. Tunapofanya vitu mbalimbali katika biology, we use sense organs, but also kuna vitu mbalimbali ambavyo tunavitumia. Kwa mfano, we use measurement ni vipimo. Unavyosema labda nataka nijue urefu wa huyu mtu ni futi ngapi, maana lazima nifanye ni measurement. Now tunaweza tukapima either mass ni uzito, tunaweza kupima length yani urefu, tunaweza kupima temperature, and even the pulse rate or heart rate. We can measure all of these things in biology. So now you can take all of these measurements in biology. When carrying out biological investigations, measurements like mass, time, temperature and length are unavoidable. Huwezi ukasema nafanya majaribio bila kujua kwa mfano uzito wa kitu. Kwa sababu unafanya majaribio unatakiwa unajua je, hichi kitu kinaongezeka uzito au kinapungua. That's one among the results which you can give a commentary on it. That according to experiment 1, 2, 3, the commentary is that the mass of 1, 2, 3 has been decreased or has been increased. So all of these measurement can be given commentary. Unaweza kuzitolea maelezo mbalimbali. We have various instruments which are used to measure different things. We discuss them in a biology laboratory later in the coming topic, but also in this topic we are discussing them a little bit. We have the beam balance, which is used for measuring mass. The beam balance, we shall see it in the biology laboratory, how the beam balance looks like. And then we have the thermometer for measuring temperature. When we are talking about thermometer, we have a lot of them. Some of them, they are digital thermometer, normally used in infants. They used to take the rectal temperature. We have the mercury in glass thermometer. We have the alcohol in glass thermometer. A lot of them, which I can't elaborate them here, but normally we use the mercury in glass thermometer for adults, and in infants who are very small and the rate of loss of heat is very high because of the large surface area to volume ratio. We normally use the digital thermometer, but some of the explanations you can't understand them now here. You'll understand them later in your Form 3 or Form 4 biology. But also we have the clock, or sometimes we call it the stopwatch, for measuring time in different experiments. Some of the experiments they require us to know the time taken for different results to take place. We have the ruler for measuring the lengths. Wenda umeshaenda kuchukua dawa za minyoo, kaambiwa simama hapo, wanakupima height, and then unapewa tablets, unapewa vidonge kulingana na height yako. We have the pulse rate. Pulse rate can be measured by using the stethoscope. Stethoscope is the device used to measure the heart rate or the pulse rate by pressing the fingers firmly on the skin. But also you can measure it by pressing fingers firmly on the skin. But this method doesn't tell us exactly what is the heart rate or what is the heart pressure. The stethoscope is the specialized device used to measure the heart rate, and it tells us the exact number of the heart pressure or the blood pressure. The study of biology, like any other subject, involves scientific processes. The scientific processes involved in the study of biology include observation. These are the stages now. In the stages of scientific process, we have observation, measurement, and experimentation. Through these processes, the study of biology becomes possible. So you observe something, and then you measure it, and then you can do experiment on it. You can do experiment to know different things regarding that. Now, in the coming session, I will start explaining the importance of every of these steps of the scientific processes, the importance of observation, how do we observe, and then the importance of measurement and how do we measure, and finally on the experimentation, how do we perform these different experiments in order to know different things in biological studies. Let's say we want to know disease ambayo patient fulani anayo. How do we know that? We shall discuss it in the coming session. So thank you, and later wish you nice studies. Let's meet in the coming session.